Good evening. My name is Beverly Ramos, and our topic is cloning, and our claim, the affirmative side, is the amount of funding should be raised for cloning research. Our first point is many people are at a disadvantage without extensive knowledge of cloning due to the lack of funding. Our second point is without increased cloning funds, we may not be able to fully exploit its benefits. And our third point is if we increase funding for cloning research, we can expand our knowledge in various medical subjects. Regarding my first point, many people are at a disadvantage without extensive knowledge of cloning due to lack of funding. According to Stephen Wilson from the West Virginia University, cloning and genetic modification are in existence because of the promise for possible benefits. So that just demonstrates that cloning exists just for the good of people. People that need organs can already have them if cloning were more advanced. And also, it's also good for infertile couples. If they're infertile couples, they can have a child if they want to. And it can also be used for various medical research, disease research, and we'll go into that later. But the second point, without increased cloning funds, we would not be able to fully exploit its benefits. I'd like to talk about cosmetics. According to Simon Smith from Human Cloning Foundation, most people are aware of the breast implant fiasco. Many came to believe that the implants were making the patients ill with diseases of their immune systems. With human cloning and its technology, breast augmentation and other forms of cosmetic surgery could be done with implants that would not be any different from the person's normal tissues. So that makes breast augmentation safer because the substance would be more natural, considering that's from another human being. In addition to cosmetics, also according to Simon Smith, with new technology, instead of using materials foreign to the body for such procedures, doctors will be able to manufacture bone, fat, connective tissue, or cartilage that matches the patient's tissues exactly. Anyone will be able to have their appearance altered without the leaking of silicone gel into their bodies or the other problems that occur with the present-day plastic surgery. For organ transplantation, in theory, the clone organ, according to the U.S. Department of Energy Genome Programs, in theory, the clone organ could then be transplanted into the patient without the risk of tissue rejection. If organs could be generated from cloned human embryos, the need for organ donation could be significantly reduced. And it would also quicken the supplies for organs because people wouldn't have to wait and they wouldn't have to wait so long for compatible organs. And also, according to Ashwini and Becker from articleswage.com, human cloning can be used to clone livers used in liver transplants as well as in kidney transplants, though cloning of kidneys. Human cloning could be used to clone bone marrow for individuals suffering from leukemia. So we know that cloning can be used to treat various diseases and cosmetic procedures. My third point, if we increase funding for cloning research, we can expand our knowledge in various medical subjects. Through copies of animals that can be used for research, we have cloning animal models for disease. According to National Center for Research Resources, much of what researchers learn about human disease comes from studying animal models, genetically engineered to carry disease, causing mutations in their genes. Cloning technologies might reduce the time needed to make transgenic animal models, and the result would be a population of genetically identical animals for study. So if we have more animals to study with, we will learn more in the medical field just because we have more, I guess, lab rats, you can say. There is also farming. Animals can be produced as resources for drugs, resources of drugs or proteins needed in order to make medicine. According to National Center for Research Resources, farm animals such as cows, sheep, and goats are currently being genetically engineered to to produce drugs or proteins that are useful in medicine. Just like creating animal models of disease, cloning might be a faster way to produce large herds of genetically engineered animals. Cloning can help produce stem cells for more research. We'll talk about that. And defective genes. We won't have to worry about defective genes anymore. According to Simon Smith, the average person carries 8 defective genes inside them. These defective genes will allow people to become sick when they would otherwise remain healthy. With human cloning and its technology, it may be possible to ensure that we no longer suffer because of our defective genes. And lastly, our plan of action is to remove federal restrictions of funding as well as instill the benefits of cloning on society. And before we move on, I'd just like to define some terms. Embryo cloning. In this procedure, identical twins are produced basically by reproducing how twins are created naturally. A few cells are extracted from fertilized embryo, which are induced to develop into duplicate embryos. The twins are thus formed to have identical DNA. Reproductive cloning. The DNA is removed from an ovum and replaced with the DNA extracted from a cell taken from an adult animal. And the fertilized ovum, which is called the preembryo, is implanted in the womb and develops into an animal. Therapeutic cloning is a procedure in which cells, typically skin cells, are taken from a patient and inserted into a fertilized egg whose nucleus has been removed. Scientists then extract stem cells from it and use those cells to grow tissue that are a perfect genetic match for the patient. Parkinson's disease, progressive disease of the nervous system marked by tremor, muscular rigidity, and slow, imprecise movement. And gene pool, the stock of different genes in an interbreeding population. And lastly, in vitro fertilization, or IVF, process by which egg cells are fertilized by sperm outside the womb.